Hello, everyone, and welcome back for Chapter 5. Here we're going to provide an introduction to a select number of eukaryotes that are important to microbiology. This will cover a broad array of eukaryotic organisms from fungi to protozoa to helminths to algae, and briefly the what is sometimes referred to as a superorganism, the lichen. We'll begin from the top with our discussion with fungi. Fungi are chemoheterotrophs, so they're able to obtain their energy and their carbon from organic matter. You may have heard of yeasts before, and yeasts are the single-celled fungi, and molds and mushrooms are the multicellular fungi. Yeasts do have cell walls, fungi in general have cell walls, and those cell walls are composed in part by a material that provides more rigidity and structure to it that we call chitin. And we covered chitin earlier in the semester. Chitin is a polysaccharide. Their method of reproduction it can be considered more complicated than bacteria. They can reproduce asexually through budding that we've spoken of, and they can also do sexual reproduction using spores. So we have our three types of fungi, the molds, the yeasts, and a third category that's a little bit of column A, a little bit of column B, the dimorphic fungi. Let's go over the physical characteristics of molds first. Molds are composed of these multicellular filaments that you can see here in A and B, which are referred to as hyphae. And hyphae may be septate, and that's where there are cell walls that separate each one of these cells here along the way. Or they can be what we refer to as, this looks like a weird spelling here, but it's pronounced cenocytic, meaning that there aren't these cellular units that separate these different cellular areas. They don't have what are called septum or septa, if there's multiple ones, and they share a cytosol. And the way these filaments grow is they grow at the tip. So every tip that you see here can have an outgrowth along the way. And additionally, if this hypha or hyphae, if you're talking about more than one, if it breaks, then you can have a new hypha that forms from the part that gets broken off. So that would be a form of asexual reproduction. There are a couple different components of these hyphae that you need to be aware of. There are vegetative hypha, and these are the ones that are going to be embedded in whatever is providing that organic matter, the medium for that organism. And then we have aerial hypha, and these are the ones that protrude outside of it, above that medium, and are used for forming spores. So in this image here, we have the vegetative hypha, which is more of this white, pinkish, yellow mass growing into this agar plate. And then if you see these little black spots, this is growing up out of the media, and it is considered the aerial hypha. Here's a little cartoon image so you can see exactly what we're talking about. We have the vegetative hypha, which are inside of the medium, extracting those organic nutrients out of it. And then we have the aerial hyphae, which grow up. So you can see this aerial hypha growing up. And then you have this spore-forming sac at the very top there. Moving right along, we can see here that a mycelium, as shown by this mold growing on this bread, is a filamentous mass of accumulated hyphae. And if there's enough hyphae present, you can see them collectively forming a mycelium visible to the unaided eye. And other examples are mushrooms. And there is a growing bioeconomy around mushrooms besides just the culinary type that we eat. For example, you can buy furniture that is made from the mycelium that mushrooms produce. And they also use it to make imitation leather. So here's another example, this MycoWorks company. Um, and that can be put into products such as purses and a lot of other products that you might use around the house. And again, we were talking about these hyphae and how they can be broken apart and generate new hyphae. And so we're going to go over the two types of reproduction here. One is called fragmentation, and this is where you have a piece that is broken off and it develops into its own fungal colony. And this is asexual in nature. And then we have the sporulation, which is where you have those aerial hyphae that produce the spores, and those spores can go off and generate a new organism. And these spores can be recombined with other spores and therefore go through a sexual reproductive pathway, or they can, depending on what spore they can just lead to a generation of their own hyphae, and that would be an asexual pathway. So in the example shown here, if this is just a single spore that had recombined with another, then this might be a asexual form of reproduction. And here we can see those sacs where the spores get formed in the aerial hyphae. And in this image, we're taking a look at the sexual reproduction of spores, of molds in general. And I like to start here with the spores here. And those spores can germinate in order to form the mycelium. And something that's incredibly interesting about fungi is when we come from our gametes, we develop a zygote, and that zygote is now diploid, which means that we have one copy of each chromosome from our mother and one copy from our father. So di meaning two, ploidy meaning chromosome. So we have two copies of every chromosome that's in our cells. Well, in the example of fungi, that's not true. Actually, the mycelium are only haploid. They have half the number of copies, 1N. And these haploid cells, they go through a process where two different mycelia can fuse together in order to form what we call a heterokaryote. So two hyphae from two different cells. They combine together into the same cell, and shown by this black dot and this red dot, you can think of those as the nuclei. And so it has a nuclei from, quote unquote, the mommy and the dada, and they combine together in the same cell. And because those aren't in one nucleus like they are when, say, humans or mammals recombined our gametes, the sperm and the egg, we form one nuclei together instead of two separate ones. So we have it all in one in nuclei, but they have it in two or different nuclei. And hetero means different, and so karyo means chromosomes. So we have different chromosomes, and this is referred to as the heterokaryotic stage. Now, from there we go through a process, and that's called the process of combining two mycelium cells. It's called plasmogamy. The process of this heterokaryotic stage forming into one nuclei is called karyogamy. That's where the two nuclei fuse together. And now we have our zygote, which is 2N. It's diploid because the mother's chromosomes and the father's chromosomes, so to speak, came together to form a single chromosome. So now within this little black dot is a copy from each parent. And now they go through meiosis, and this results in a haploid spore being formed. And those haploid spores can germinate and become the mycelium once again. Now, in asexual sporation of molds, it's a lot more simple than this. You have your spores that can germinate and become mycelium. And they're going to replicate more mycelium cells and grow into a longer hyphae. And through mitosis alone, so not meiosis, just through mitosis, they can generate spores as well. And that is a form of asexual reproduction. So what are we lacking here? Well, we're really lacking plasmogamy, karyogamy, and meiosis in that process. Okay, so that covers molds. Let's discuss yeasts for a little bit. Again, they're single-celled. They're non-filamented. There's no hyphae because, again, they're just single-celled free-living organisms. Here's an example how yeast cells typically reproduce. They go through a process called budding. So here's the mother cell, and there's an outgrowth of what we call a daughter cell forming at one of the ends of the cell. So that bud is initiated by the parental cell, and the parent cell's nucleus then divides into two, and the nucleus is then shared across, or the nucleus migrates into that bud growth there. And the cell wall is synthesized as well as other components, intracellular components, until eventually it becomes large enough that it and has all of the essential ingredients that it breaks away from the parent. And that leaves behind, so here's a bud forming, here's the mother cell, here's the soon-to-be daughter cell, and that will actually leave behind where that septum forms a budding scar from a prior generation. So as this mother cell divides, you can see it's done here, here, and here. There's going to be a scar there, and there'll be a scar there if that continues to grow outward into a full daughter cell. I said there was 3 categories. And the last category I said was a little bit of both. And these are called dimorphic fungi. So they can have 2 bodily forms here. They can be, they can grow as either as yeast or molds, and it depends. It specifically wants to grow as a mold or a yeast depending on the particular environment. So on certain temperature, CO2, or concentration, it'll switch between the 2. Dimorphic fungi are ones that we're really concerned with. The pathogenic fungi out there usually have dimorphic characteristics, and when they're causing certain infections, they may want to be either yeast-like or mold-like, depending if they're growing inside of someone's body or growing out in the environment. And that was a pretty short discussion of fungi. And we're going to now cover the protozoa. So here's an example of a protozoa here. They are single-celled. They're incredibly diverse. Like many eukaryotes, they are also heterotrophic, so they consume organic material in order to obtain their energy and carbon. And most of them are non-pathogenic, although there are some that are parasites to humans and other hosts. And before we continue with this discussion, let's briefly talk about symbiosis and symbiotic associations. Symbiosis just simply means living in some close association, having some sort of a relationship with another organism. And there are 3 main types of symbiosis. One is called mutualism. And mutualism is where both individuals benefit. For example, E. coli that live in human intestines, they have a nice home to live in. We provide them with all the food they need and warmth and all those wonderful conditions. And they might synthesize some essential components that we need. For example, vitamin K is synthesized by bacteria in the gut. So we can see here that that's mutualistic for both of these organisms. Both of them benefit. So 2 positives there. And commensalism is where one benefits, but the other is it's kind of neutral to that effect. So it's not a negative or positive to one of them, and one of them benefits. That is commensalism. And a great example of this are the Demodex mites. that actually live in your hair follicles. They live in around your eyebrows and your eyelashes and in your hair and so forth. And they live off of the detritus material, the skin that's dead skin that's flaking away. They live off of that. And it's not harmful to us. It's not a positive for us. It's not a negative for us. It doesn't affect our survivorship at all. And so we would say that that's a commensalism relationship. Mutualism, commensalism, and then parasitism. Parasitism is close to commensalism, but instead of being a positive for one and a neutral effect on the other, parasitism is where it negatively impacts the other. This would be a parasite host relationship. So the parasite is the one that benefits, and the host is the one that harms. An example of this is going to be the protist that causes Giardia, Giardia intestinalis. This organism lives in the intestines of mammals and can cause a diarrheal disease. It obtains its nutrients and all the good stuff, just like E. coli does from its human host, but it does cause disease in that host as a consequence. So it's a positive for one and a negative for another, makes a parasitic symbiosis. Now, parasites have very complex life cycles, and they often have more than one host that hosts, no pun intended, different parts of that life cycle. You can have a definitive host, and the definitive host is where that parasite reaches maturity and can reproduce sexually. So there's two components to it. It has to reach maturity, and if that parasite can reproduce sexually, it can do so in that host. An intermediate host is a host that a parasite infects and is in a developmental form. So it's not in its fully adult form, capable of reproducing sexually. One organism that has a parasitic relationship with humans is the Entamoeba histolytica protozoan. This particular protozoan is able to form something called a cyst, sometimes called a capsule, but I prefer not to use that word because it may be confused with bacterial capsules, which are totally different in the way that they're synthesized and their structure, but they serve kind of a similar function. A cyst stage is really important because when it is formed into a cyst, this particular organism is resistant to poor nutritional environments, poor moisture environments, so drying out, low oxygen conditions, poor temperature conditions, so on and so forth. And the reason why these organisms do this is it's a great stage to be in when you're trying to hop from one host to another, where environmental conditions vary and they're not always perfect for you. Entamoeba histolytica is an organism that's very commonly causes something called amoebic dysentery, since it's considered an amoeba and it causes dysentery. Dysentery is a term that we'll use in this semester here and there. It's an inflammatory disease of the intestines, and it usually comes with severe diarrhea. And as a consequence, it can be bloody or have a lot of mucus present in it. And so this is not something that you want to get. Okay, and then there are quite a few select protozoans to cover. We're just going to pick on a few here: the amoeba, the paramecium, Giardia, and trypanosoma. Here are the amoebozoa. Amoebozoa are actually really cool in that they move using this pseudopod structure. And you can see it in this GIF here, the pseudopod. Pseudo means false, and pod means foot. So it creates what scientists call a false foot. And so it'll protrude out a tip of their cell plasmic structure attached to the surface, and then using its cytoskeleton, it draws in its intercellular components and expands that area out therefore pulling its rear towards that area. Some of them are parasitic. So we talked about E. histolytica on the prior slide, causing amoebic dysentery. And then another one is Balamuthia, which can cause an infection of the brain, which can lead to amoebic encephalitis. Itis means inflammation, and encephala would mean, around your brain, encephalo. Some of them are what we call cellular slime molds. These are typically not harmful organisms. And I want you to keep in mind here that we're talking about amoebozoa. We are already covered yeast and molds. These are not molds. They were it's kind of a misnomer where we categorize them as molds. The reason why it was categorized as slime molds is they formed this structure here that sort of looked like the aerial hyphae that we discussed earlier, but actually these are not molds at all and quite unrelated. And they can aggregate when they're under unfavorable conditions and form this structure here in order to try and relocate to greener pastures. And I just wanted to share with you what this looks like. So these slime molds are considered single-celled organisms, but they come together and form a quasi-multicellular organism. And these quasi-multicellular organisms can actually go around and hunt other cells if they eat other cells or search food out in order to consume them. And this is kind of a stepping stone to multicellularity, these individuals working together as a sort of pack. So there's some sort of environmental trigger, maybe an autoinducer involved in this, and it's bringing these cells together. And what's really unusual, as it says here, they participate in something called self-sacrifice. So for some reason, some of these organisms are willing to give their lives and resources in order to allow others to survive. So let's go ahead and watch that play out here. So the cells that make up the stalk, they will not live on after this event. But those that are up here in this sort of what we might call a fruiting body, and we'll see what happens here in a moment, they can go on to make other progeny. All right, what's not shown in that clip is what they do after they've gone on to make other progeny. So these cells, eventually this tip will explode, shooting the cells across and hopefully reaching other areas that are better environmental conditions so that they can continue and survive on. And I think this is probably just the same clip here. Let's see. Yes. Okay, let's switch gears now and let's talk about the paramecium. Paramecium are unicellular ciliates. And why are they called ciliates? Well, if you think back to chapter 3, the cell, we learned that eukaryotes have a structure that's unique to them, and that is the cilia. Not all eukaryotes, but prokaryotes don't have cilia. And cilia are these hair-like protrusions from the cell. In fact, that's how I kind of remember that they only belong to eukarya and not prokarya. Prokarya do have fimbriae, but they don't have these cilia. And these cilia present on this paramecium are great. They're used for propulsion, so they can allow it to have a swimming action at incredible speeds. And also they can alter the current in the fluid that they're present in order to draw food in so that they can consume that, whatever organic matter it is. And so they're named after the cilia. And I just want to clarify, they are hair-like, just in the way that they look, not in the way that any sort of their components or structure or their evolutionary origins. They are not hair. Fortunately, none of the paramecium are known to cause human disease, but they are very important to scientists because we can study them to try and have a better understanding of how reproduction works. So they are very important as study specimens. All right, here is Giardia, and Giardia is important to talk about because it is the most common human intestinal protozoan parasite in the US, bar none. It's present on basically every waterway you can imagine. This is the chief reason why you don't want to drink water when you're out and about on a hike, because there's a good chance that Giardia is lurking around in that water. Giardia are pretty nasty little beasties. They have this sucker disc, and this is what they use for attachment to the intestinal lining. And instead of like E. coli, where E. coli gets its nutrients from whatever's floating around in your intestines, they're working on getting nutrients right out of your intestinal cell wall itself. Um, and they are secreted in a cystic stage, and they go through the feces. And that cystic phase is very tolerant to inhospitable environmental conditions, which makes them so resilient in waterways and present just about everywhere. So that is Giardia intestinalis. Now let's talk about trypanosoma. Trypanosoma is a blood parasite. It's transmitted by insect bites. This actually was one of the, we do have a copy of these slides in the lab, so hopefully we'll be able to take a look at that this semester. They have, as you can see here with this blood sample here, they have these very long bodies, and they propel themselves by undulating the membranes. There are two main diseases that we need to talk about with these two, and these are considered emerging diseases, so they're becoming more common in the world, and that is Trypanosoma brucei, and that causes African sleeping sickness, and then Trypanosoma cruzi, which causes Chagas disease. This Chagas disease is something that is of some concern to us in Arizona. It can be transmitted through this organism here called a kissing bug. This particular bug, if you'll find it in backyards all over in the Arizona area. So far, it hasn't been a big issue as far as people getting bitten and receiving Chagas disease, but it's something that the CDC is definitely trying to keep their eyes on. All right, so that covers fungi and protozoa. Let's go over the helminths next. Helminths are multicellular, and they are considered animals. So we're getting a little bit more complex here. They produce eggs, and they have larvae, and these are the microscopic components of our helminths. But they also have adult forms, which may not be microscopic entirely. They're a parasitic species of helminths, and they have complex life cycles, as do most parasitic organisms. And what you're seeing before we talk about dioecious, monoecious, just looking at the image here to the right, what you're seeing here are tapeworm images. So they do have this little hook here that's how they attach themselves to the intestinal wall, and similar to Giardia, they feed on the intestinal wall. Something else that they have is this concept of being monoecious or dioecious that we'll cover. They can be dioecious, which means you have male and female reproductive parts in different organisms, or they can be monoecious, so they can be both male and female reproductive parts in the same organism. Monoecious, to be more exact, means that you're capable of producing both male gametes and female gametes. There are a variety of different helminths. There are the two broadest categories are flatworms and roundworms. And yes, that's exactly what it sounds like. Flatworms are flat. So for example, the tapeworms that I showed in the last image, and then roundworms are round. They have more round bodies. And we'll talk about two subcategories of flatworms, the flukes and the tapeworms. Starting with the flukes, here we have Clonorchis sinensis, and this is like a liver fluke. They have leaf-shaped bodies. So if you imagine a leaf is kind of shaped like this, kind of pointy on one end and kind of more roundish, but still a little pointy on the other end. And these are flat if we were to look at it from a side view. They have their ventral. They have a ventral oral suckers, and these actually hold the worm in place. So down over here, the ventral side. They're still pretty simple organisms. They lack a complete digestive system, and so they need to absorb their nutrients directly from the environment. They don't digest it entirely on their own there. Again, like a lot of these parasitic organisms, they have a highly complex life cycle. And what you need to know about flukes is and like this liver fluke here, they are monoecious. So they're hermaphroditic. They're capable of generating both male and female gametes. Here's another flatworm called the tapeworm. That sucker with the hooks on it that I showed you in the previous slide, that is called a scolex. They range in size from as little as 1 millimeter. Again, that's about 10 sheets of printer paper to as large as 10 meters in size. They, like the flukes, they lack a complete digestive system, so they need to absorb those nutrients straight from their surroundings, from their environment. The different segments of their body are called proglottids, and these proglottids, that are produced, they can live as long as the scolex is still alive. Now, roundworms, since we discussed flatworms already, they range in about 1 millimeter up to 1 meter in length. They're actually the most abundant animal group on Earth. About 4 out of 5 animals on Earth are a type of roundworm called nematodes. And so they live basically in all environments where you find animals. And only a select few of them are actually parasitic, but they are important to be aware of. They possess, unlike the flatworms that we looked at, they can possess a complete digestive system. And most common cause of chronic infections in human diseases may be the result of these flatworms. As I said, they're incredibly common. And you can see in this little snippet that I took from N.A. Cobb, who studied flatworms, he said, in short, if all the matter in the universe except the nematodes, which again are a type of roundworm, were swept away, our world would still be dimly recognizable. And if, as disembodied spirits, we could then investigate it, we should find its mountains, hills, valleys, rivers, lakes, and oceans represented by a film of nematodes. The location of towns would be decipherable, since for every massing of human beings, there would be a corresponding mass of certain nematodes. Trees would still stand in ghostly rows representing our streets and highways, the location of various plants and animals. I'm sorry, my computer paused for a sec there. Would still be decipherable, and had we sufficient knowledge, in many cases, even their species could be determined by an examination of their erstwhile nematode parasites. We must therefore conceive of nematodes and their eggs as almost omnipresent, as being carried by the wind and by flying birds and running animals, as floating from place to place in nearly all the waters of the earth, and as shipped from point to point through the civilized world in vehicles of traffic. This slide is going to cover several roundworms that do cause infections. This includes Ascaris lumbricoides, which infects over 1 billion people. Pinworms affect about 11.4% of the US, including about 30% of children. I think these numbers might be a little bit outdated, and hopefully that's been declining, but pinworms are very prevalent in the American Southeast. Whipworms infects about over 1 billion people. Threadworms infect 30 to 100 million people, and hookworms infect greater than 500 million people. So, as you can see here, they're very common pathogens around the world. Here are some pictures to show what's the different types of diseases that they can cause. The first one in the upper left is guinea worm. This infects just the subcutaneous tissues of skin and limbs, and it can lead to painful ulcers. This particular one is actually near eradication, and this is due to clean water initiatives. This is you pick it up if you go into water that's contaminated with a guinea worm, and so we're making excellent progress on that front. Loa loa is it's another name for it is the African eyeworm. It infects subcutaneous tissues, especially around the eyes and skin as well. And the middle one you see there is hookworm. So we talked about hookworms on the previous slide, and that infects subcutaneously as well. And the last one, Wucheria bancrofti, this particular roundworm infects the lymphatic system, and it causes something called elephantiasis, which you can see in the leg of this particular individual. So it can cause severe limb swelling, and this one is spread through mosquitoes in tropical regions. Okay, so that's our coverage for helminths. Moving right along, we're going to look at algae. And you don't need to memorize this image here to the right, but it's going to allow us to talk about one of the features of algae. Unlike everything we've discussed so far, algae are autotrophic. So they can get their energy and their carbon. They can get their energy by using sun's photons of light to split water molecules, and they can get their carbon from inorganic sources or CO2. Their full name would be photoautotrophs, and they can be unicellular or more multicellular, and they can live in freshwater environments, marine environments. That just means in the oceans and ocean-like regions, and they can live in terrestrial environments as well, so up on land. Algae, if we exclude cyanobacteria from algae, oftentimes textbooks will miscategorize cyanobacteria as algae. Sometimes they call them microalgae. But let's make a distinction here that algae are eukaryotes and cyanobacteria are prokaryotes. They're bacteria. And that is the biggest difference, the most broadest difference we can categorize organisms are eukaryotes and prokaryotes. So it just doesn't make any sense putting them in the same category. But if you exclude cyanobacteria, algae are still very important. Our main photoautotrophs are going to be algae, plants, and cyanobacteria. And these guys alone they synthesize roughly 40% of Earth's oxygen, and they fix CO2 in the organic matter. So about roughly the same in organic matter as well, and in aquatic ecosystems. And so they're very important ecologically and essential. That's mainly why we cover them here, except for one other thing I'll get to. And that will take a moment here. But first, let's just outline that they do have something, a organelle in them called a chloroplast. And that chloroplast, much like the mitochondria that we discussed, the chloroplasts are originally came from prokaryotes, in particular the cyanobacteria. An algal-like organism consumed cyanobacteria through something like phagocytosis, for example. And the cyanobacterium was not killed inside of it, but took up a permanent residence inside of that cell. And over time, it evolved to become the chloroplast, which is the photosynthetic, the organelle responsible for doing photosynthesis. What's so weird about these chloroplasts is, unlike mitochondria, which have two membranes, makes a lot of sense, just kind of like a bacteria does, like a gram-negative bacterium. Chloroplasts can have 1 to 4 membranes. And there is a hypothesis that this might be the result of secondary endosymbiotic events, where another membrane was obtained from that secondary endosymbiosis event. So I'm going to take you through that. Again, you don't have to memorize this chart, but I think that this is really cool to show how algae came about land plants, and then other specific types of algae. So let's start here with the beginning here. What we have, it says it's a heterotrophic protist. So this is a single-celled organism, and we can see our mitochondria is already present. It's a eukaryote. And somewhere along the line, it took in a free-living cyanobacterium, probably through something called phagocytosis, where one cell eats another cell. And maybe it made a living like that. Maybe it hunted these cells and consumed them as a source of food. But one time this happened, and that cyanobacterium was not degraded. It was able to survive and live inside of that cell and was able to reproduce with that cell over and over again, and eventually became a chloroplast here. Here it's labeled as a plastid. A plastid is just a name for a chloroplast or a chloroplast-like organism. And this is where we have a fork in the road, and different types of organisms evolved. We had the red algae that evolved directly from this event. And then we further had what formed were the green algae and land plants. So algae and green algae and land plants are very closely related here. But like I said, some of these chloroplasts have even more membranes than you would find in, say, a mitochondria. They might have up to 4 membranes that surround that organelle. And scientists believe, they have very strong evidence for this, that this was the result of another endosymbiosis event, a second one. So here we have our algae here. Great, we've got our mitochondria, our plastid. Here's the main chromosome nucleus. So these 3 different sources of DNA in all 3 of these. This particular organism was ate by some other heterotrophic protist that had no business doing photosynthesis. And that cell took up permanent residence in there, and you can see the extra layers of membranes around it. And this organism went on to do all sorts of amazing things. And if you ever take Bio 182, you'll learn about these different organisms, but led to this giant evolutionary expansion to many other organisms out there, some of which don't even do photosynthesis anymore. So I'll leave it at that with our discussion of algae and evolution, but just an amazing journey when you think about it. And one other thing I will mention about algae, just their general traits, is they can reproduce both sexually and asexually as well. Okay, you're probably wondering, what is this image of a food label up on my screen for? Well, some algae are edible, and they can be a source of, say, example, the seaweed that you find with, if you ever have a sushi roll, that outer green layer is made up of algae, but also a very important chemical called carrageenan. And this is a thickening agent. If you look here, if you look at the back of a lot of yogurt products, if you look at the back label, you might see carrageenan on the back label, and that's what can help thicken up that yogurt. Nowadays, a lot of yogurts are actually made in a giant vat, and when yogurt is initially made, it turns into the solid mass, as we all know and love yogurt for. But then they inject it into whatever vessel it is that is being sold in the store, so like your little plastic container. And once you break up that yogurt, If it's completely done fermenting, you're not going to get that solidification again in the same manner. Just like if you take yogurt, you put it in your bowl and you mix it up, it softens up and becomes a little bit more liquidy. The same thing happens. And so what food scientists do is they add a little carrageenan and that helps firm it up. And then, of course, from algae, we get that polysaccharide agar, which we use in our agar plates in our microbiology. Um, Other things that we need to know about algae is there's different types. So we have brown algae, which make what we call these kelp forests or kelp. So here we can see in this image these giant multicellular algae. Again, not all of them are unicellular, forming these multicellular structures underwater, and they almost look like they're on their way to becoming terrestrial plants. I mean, they have things that resemble, they are not, but these structures that resemble leaves. And you can see that there's sort of stem-like structures here, and things that resemble roots. They're called holdfasts that hold them solidly into the ground or on rocks or dirt and whatnot. Although they do lack true plant tissues, they're not considered plants. They're still algae. And we have another group of them called the diatoms. I wanted to share diatoms with you because diatoms are very important that they make silica-based cell walls. So silica, it's an element from the periodic table of the elements, and it's a glass-like particle. If you think about glass, it's made from silica. And these little components, they've built up on our planet over millions of years. And these little pieces of, these sort of like exoskeletons of diatoms create an excellent source of these glass-like compounds here on Earth. And we use them for filtration devices, and we use them also in our gardens or maybe around the kitchen. Another name for this is diatomaceous earth. And these are the skeletal remains of these diatoms. These are algae, even though they have this crystalline silica outer layer to them. And they come in many different morphological shapes and structures that are really just amazing. And I wanted to share those with you. And these pieces of glass, the remains of them are silica, are very sharp and brittle. And what they end up doing is through a non-chemical action, just a physical action, when you have a insect that has a chitin exoskeleton and it crawls across all of this broken glass, it actually shreds up their exoskeleton and kills them. So it's a non-chemical induced insecticide, which is a great product to use around the house. Okay, and then finally, just one last detour here. I wanted to mention lichen because it really ties in a lot of the concepts that we discussed here. We discussed fungi and we discussed algae and symbiosis. And what is lichen but a mutualistic symbiosis between fungi and algae? And you see it just about everywhere in nature. Each one of these images here are an example of lichen. Whether you see this pigmentation growing on a rock or you see it on trees, they can be lobate in nature, they can be kind of powdery looking in nature, or they can kind of look foliose, or they look kind of like more like leaves or bushes, more like bushes, things like that. Lichens, it's pronounced lichen here in the US, they're communities of fungi and some sort of photoautotroph, either cyanobacteria or algae. And when they come together, we call this a superorganism. And because they act as like a single organism, even though they're two distinct species, or could be multiple species living in there. But you always need to have a fungi and a photoautotroph. And the way this symbiotic relationship works, remember this is a positive for both groups there, is that the algae or cyanobacteria, the photoautotroph portion, they are the source of organic carbon and energy. So they're going to take those light photons of split water and harvest energy and make ATP from it. And they're also going to make organic matter by harvesting CO2 and fixing it. So they are providing the source of energy and the source of organic carbon for the fungus that are heterotrophs. So they're chemoheterotrophs. They have to get their energy and their carbon from something else that's already fixed that carbon and already produced that energy in a usable form. And the fungi provide other things to the algae and cyanobacteria. So they get their nutrients from the algae or cyanobacteria, but what they can do is they provide this home for the algae and cyanobacteria that take up residence. They are able to better harvest moisture, lock in that moisture there. They produce waxes that can hold in moisture. They provide a protective, cozy little area for that algae or cyanobacteria to do its thing and do photosynthesis for them. So I thought that would be a great way to tie in those different features of those eukaryotes. That's the end of this lecture. Let me know if you have any questions. Thank you so much for joining in.